Luigi Naldini on this, partly because I know that Genenta is working on the lentivirus system and has been, and you in different spheres have been working on lentivirus for a long time, maybe back to the mid-90s. I may not have counted all the companies, but I've got at least three. You've got Genenta that you're with now. You've got GeneSpire, which is another San Rafael spin out in the rare disease area, and there was Cell Genesis. There have probably been many in between. But I guess that the different, can you say a few words about the difference between, that pioneering time, not that you guys aren't all pioneers, but, pioneering really at the start of gene therapy, and what we're able to do now and how that changes the picture for the capability of delivering gene therapy to a broader audience?
Yeah, thank you. That's a good question. I think originally gene therapy was, of course, a great promise, but really the challenge were the vectors to achieve safely and effectively gene transfer into the desired target. And the first generation of system that we have been using were either not very effective or potentially more dangerous, giving rise to like either inflammatory reaction or a genotoxicity. I think the big changes, which is really is the reason for the recent excitement driven by clinical results, is the change to a new generation of vector, which have been there for no more than 10 years already in the clinic. A lentiviral vector represent, I would say, the key system for integration. So for making a permanent change in the genetic information of a cell. This is essentially if you treat a proliferating cell, like a stem cell or a T cell, which need to pass on the genetic modification to the progeny. And AAV, as we just heard, for a mostly non-integrating transfer into a stable tissues. Now, these two platforms have a wide range of activity. Lentiviral vector have been primarily tested until now into hematopoietic stem cell and lymphocyte. As you mentioned, they are far ahead in Europe, but also in the US now. There are several approved drugs in stem cell and T cell gene transfer. There are many more to come. They are the key driver for genetic disease, on the one hand, mostly in stem cell therapy, and for cancer, the CAR Ts exploit lentiviral vector as well for modified lymphocyte. So this is the level of validation in stem cell gene therapy. There are probably more than nearly 400 patients treated in 5 or 6 different diseases today. So that's where we started from. If we can now go to what we are doing now, with Genenta, is actually leveraging on this experience for really validating a new concept, which is gene-based delivery of immunomodulatory cytokine to tumors. So treating tumor, of course, you cannot fix a gene. You need to leverage mostly at this point of immunotherapy. So either you use CAR T to try to fight a single antigen in the tumor, or you try to activate the immune system against the tumor through cytokine, like interferon and other ones. The problem with cytokine delivery is that conventionally, these are given like recombinant molecules. They have important effect throughout the body, and they may also become neutralized at certain point. What we are doing, Genenta, is using the cell and gene therapy to deliver cytokine from within the tumor, exploiting cells which are recruited to the tumor for local delivery of cytokine. And
that presumably requires sort of targeting within the tumor as well as targeting of the cells to the tumor.
Exactly. There's 2 levels of targeting. One is the cell which goes in the tumor, which in our case is an inflammatory cell. So these are the myeloid, the macrophages, which any tumor, essentially of every type, recruits and needs to grow. And we modify the progenitors of this monocyte, so they are carrying the gene, like for interferon, mostly in our case. But that gene is off, essentially, until those cells reach the tumor and activate the program typical of tumor-associated macrophages. And this cytokine essentially counteract the activity of this inflammatory cell, which are pro-tumoral to begin with, and they reprogram the microenvironment to trigger antigen presentation, to inhibit angiogenesis, and to mount an adaptive response against the tumor, which is in preclinical. Several models we have been using have been extremely efficient. And we have a control on that because we can control the dose of the cell which reach in the tumor. They are not going to amplify themselves. So you establish, so you have a sort of confidence level, and expression from within the tumor has a huge advantage. You can think if you do a cytokine administration, you have like peak and trough. So you reach a region where there is too much activity, maybe toxicity, and then you have no activity. And this is eventually desensitize the host. While if you have a stable output within the physiological range at the disease site, you are sparing the off target and you are achieving more efficacious activity. This is an important principle that we are establishing in our clinical trial, which has started in glioblastoma as a first indication.
So in a way, it's the opposite of some of the problem with the glycosidase diseases, that you want to deliver everywhere in those, but in your case, you want to deliver very specifically.
Exactly.
Thank you. There's a question actually that's come in that's relevant here, which is to do with attitudes from established industry, and it's also probably paying into the payment equation as well. The question basically is, do you think that big pharma sees gene therapies as a threat to well-established and profitable chronic therapies? That may be particularly the case in areas like hemophilia, I guess, but probably less so in oncology and ophthalmology. What are we hearing from big pharma, or what do we think the attitudes are? And what are we hearing from the medics in ophthalmology and oncology? Luigi, did you want to come in?
Well, I think the driver here is really the potential benefit. I mean, when we are dealing with gene therapy, we are expecting fundamental benefit from a single administration of the drug. This is often is the only chance you have for the administration, and you expect durable, transformative benefit reaching up to cure in some severe disease. This is nothing which is similar to what you can achieve with conventional treatment. You are capturing fundamental processes like genetic information or even cellular behavior, like in cell therapy. There are stem cell or there are T cell, which are, so it's a completely different model. And obviously, I don't think that the big pharma has a chance there not to plug into this. This is just happening. The data are driving it. Obviously, there are challenges. We were the first to bring, together with GlaxoSmithKline, the first stem cell gene therapy into the market. And of course, the first time we talked with them, the CMC person who was dealing with that, I said, how can we sterilize the product? I said, well, you better not sterilize, because if you sterilize the product, you're going to kill it. But on the other hand, now we have learned to do it, and we are seeing that, moving to the cancer field, when of course there are many more patients than in the genetic diseases, this become industrialized and cost will be managed, and this will become more and more a platform which can be adapted to on-site delivery. So this whole thing has to be learned, but I think it's an area for massive development, given the potential that you have. Cell become deliverer of therapy, and therapy is into the genetic basis. This is something we never had before, and of course we have to exploit it. But the cost will, it has been mentioned before. The cost at the moment, especially for the genetic diseases, obviously it's huge, it's tremendously high. But on the other hand, it's a value that you can ensure, because you save on the cost of just the conventional care for a patient who would have a dismal expectation. So if you go into the more common disease like cancer, cost will be adapted to the increasing larger. So I hope this will be, and I expect this will happen. More and more will become part of the routine. We are learning there.
In the case of oncology, you expect, or at least you expect pharma to come on board rather than just people. I mean, we know Novartis is already their
You know,
RA really worked for them.
CAR-T have been brought. I mean, you see the massive. There are thousands of trials on CAR-T at the moment. There's so many. And this is mostly only happening in the hematological diseases because CAR-T are working there very well, but they are not working so much into the solid tumor because, again, the microenvironment, as I said before, is sort of hard, essentially fighting against the T cell. Hematologists have been easy to, of course, take the CAR-T strategy because it was really belonging to their capacity. The solid tumor field needs to learn about that, but it is there. I think it's a matter of, in that case, we have to surpass some biological issues, of course, but if CAR-T becomes effective in solid tumor, this is going to happen. This is going to be a transformative change for the whole oncology field. And there's some indication that we will get in there by manipulating the microenvironment, whether through gene-based delivery, whether through other drugs. So I think that's what I expect to see.
And you expect that the gene therapy could become an earlier line therapy? Just briefly.
I think it's very possible. You see immunotherapy, how quickly it became a third pillar of cancer treatment. Gene therapy may empower. At the moment, in case of cancer, it's a form of immunotherapy. So it may really empower immunotherapy to become really powerful because you modulate much more the tumor, the fighting tumor aspect. So yeah. I think it could.